This is the second video on inheritance of traits, accompanying the first video, which I recently posted. I want to make sure I'm very clear about what we need to know as far as this unit and how it relates to everything that we've talked about in the articles and other material that we've seen over the past few weeks. We mentioned the word pedigree. Now, a pedigree is a diagram that follows the inheritance of traits from one generation to another. We did take a look at this previously, and we can see that the pedigree here, this is something that didn't actually show up as I expected it to, but we see a parent generation, we have a first generation, we have a second generation. In a pedigree, generally males are squares, females are circles, just by convention. And then when we have some sort of a trait, we can see it, how it's expressed. You would notice that this particular trait is very common in males, not so common in females. We'll talk about sex linkage a little bit later. The P generation would be the parents, the ones who are first considered in a pedigree. And then from there we get to the F1 generation, which is this group of brothers and sisters who end up marrying, and then we have the F2. That's what I wanted you to see. I'm not sure why it showed the highlighting before it showed the words. But that is what you need to understand about a pedigree. We trace the occurrence, the phenotype of a particular trait through different generations. We're looking at the phenotype here because it's a physical representation of, or a biochemical representation or behavioral representation of a particular trait. The genotype is what we try to determine after we look at the phenotype. Now, in this particular case, this looks very much like a sex-linked trait, which I'll talk about a little bit later on in this video. Pedigree charts can show us which traits are dominant, which are recessive, and also some other possible genetic means of inheritance. What you would want to know first is that diploid organisms have two sets of each chromosome. We have a pair of each chromosome. We have two copies of each gene. Those genes, if they're expressing some sort of a phenotype, they are called alleles. In this case, we can see someone who has one allele for earwax. They are homozygous, meaning they have the same gene for making earwax on both chromosomes, whereas the gene for eye color, we see two different alleles, one for brown eyes, one for blue eyes. So we have a homozygous situation here where we have both alleles identical, and then we have a heterozygous condition here where the traits are, the genes are different for the trait. Mendelian Laws of inheritance were developed by Gregor Mendel. You remember he was the monk. He studied pea plants primarily. He came up with two very important laws, which have been modified a little bit over time. If you would know that Gregor Mendel lived in the 1800s, we didn't have a great understanding of what happens inside of the cell at that time. But what Mendel did determine was that in many organisms, the pea plants that he studied, which are also diploid, just like humans, he was able to see that alleles would separate and would assort themselves independently. So what this basically means is you have two alleles that a parent could pass on to their offspring. Only one of those alleles ends up going to the nucleus that will become a sperm cell or an egg cell, so that we have one copy of each chromosome in the gametes in the sex cells, which will then eventually spread and meet the corresponding sex cell, the corresponding gamete, so that one pair of chromosomes from the father will pair with one pair of chromosomes from the mother. In the parent are separated out, so we have pairs of chromosomes, this chromosome, that chromosome. They end up becoming just, instead of a pair of chromosomes, a single chromosome in the sex cells and the gametes, which are then used to fertilize another gamete in another from another organism, and therefore you get the two sets of chromosomes coming back together with a new combination of traits. So the law of segregation would tell us that alleles are separated. You have two alleles. You have blue eyes and brown eyes. They get separated. You may either pass on the brown-eyed allele or you may pass on the blue-eyed allele if you're heterozygous. If you're homozygous, of course, you're only going to pass one copy of the allele and it's going to be the same allele. The other idea is independent assortment. So alleles for traits don't necessarily go together. So you might have brown eyes as one allele getting passed on, and ability to roll your tongue as another allele getting passed on. The idea was that they all assort themselves randomly. The tongue rolling genotype is not linked to the brown-eyed genotype. The tongue rolling allele is not linked to the brown-eyed allele. They would each randomly assign themselves to a gamete. But this really does not occur in all cases. It only occurs when alleles are on separate chromosomes. Mendel was very lucky that the pea plant, all of the traits that he was looking at, all of those traits were on separate chromosomes. So he could come up with this law of independent assortment. If two genes had been on the same chromosome, then he would have seen two alleles traveling together, because if there's two genes on this one chromosome, they would travel along and they would still be stuck together on that chromosome, and there wouldn't be necessarily an independent assortment of all alleles in that particular case. Now, during class, I showed you my leopard geckos. I want to tie this to my own experience breeding leopard geckos. If you become a animal breeder, and there is some money to be made in that, not enough to not have a regular, 9-to-5 job, but if there is a way of, making some money, if you do the pairings right and you understand the genetics of the reptiles or whatever other animals you might be breeding. For about 16, 17 years, I've been breeding leopard geckos. They come originally from desert areas in India and Pakistan and Afghanistan, but they're much more common. The ones that people raise in the United States are actually from India primarily. The dominant trait, looking at any organism, but in this case looking at leopard geckos, is the trait that if you have one copy of the allele, remember we are talking about two alleles in a diploid organism, and leopard geckos are diploid organisms. If we pass on one copy of the dominant trait, we see that phenotype expressed, and that would be the case whether they are homozygous or heterozygous. You might have a dominant and a recessive allele, but the dominant is the one, as you can see, whether it's in the homozygous state or the heterozygous state. So this particular leopard gecko maybe does have a gene that would code for something other than wild type coloration, but you would not know it because it's completely hidden by the fact that it has a dominant allele. These spots that are kind of a dark brown color is a dominant trait in leopard geckos. The recessive trait in this particular case would be being albinism, as it's called, albinism or albino, where they don't make the pigment that causes those brown spots. So they maybe still have yellow, which is part of their natural coloration, but they definitely don't have any of the brown spotting. This is a recessive trait. This particular leopard gecko might have the allele for albinism, but you would never know it because it's a recessive trait and not a dominant trait. Both parents would have to pass on an albino allele for the organism to be albino, and they would have to be homozygous for that allele. You can't have a heterozygous individual with albinism and normal coloration and have it come out as albino, because albino is recessive. It has to be two albino genes in order for albinism to be seen. Albinism, as you can see here, as I pulled some pictures together, albinism is common in lots of vertebrates. So we can have mammals like mice that are albinos. We have amphibians like the toad that is albino. We have catfish that can be albino. We have even vultures and axolotls, these salamanders that can be albino. Pretty much every species has the possibility of being albino among the vertebrates, although most albinos do not live very long in the wild. They have a much better chance of surviving in captivity because they're protected from predators, but most of these species would be very much a target for predators because they're much more easily seen. They don't have any camouflage coloration. And as I mentioned, albinism is common among the Guna people of Panama, where between 5 and 10%, about 1 out of every 7 children born is actually an albino. So this is a very common trait in some parts of the world, especially where there's a small gene pool. The Guna people, there's not a tremendous number of them, so a gene like albinism can show up kind of frequently because a lot of the people carry the trait, and since there's not a huge population, you see it end up pairings, people end up showing up with this trait fairly often. To know about how the first albino leopard gecko appeared in captivity, and it actually made a lot of money for certain people, you have to understand that the albino has those 2 recessive alleles. In this case, I just call them DD for not having dots. If I called it A for albino, well, really, the recessive condition is albino, not the dominant condition. So I'm calling the D having dots. They have the spots on them, and that's the dominant trait. That's why I have this as a capital D. And the recessive trait, small d, is not having dots because they can't make this pigment called melanin. In the P generation, the original generation, we have normal colored leopard geckos. Leopard geckos have nearly the same number of chromosomes as humans. We have 46, they have 38. We have our sex determined through chromosomes in most cases. However, leopard geckos do not have sex chromosomes. Leopard geckos, it depends on the temperature. So if you have a low temperature for leopard geckos, they tend to be female. If you have a higher temperature, they tend to be male. And then if you have a in-between sort of temperature, maybe about 80, 82, 83 degrees, you'll actually get a mix of males and females. And what's being seen in a lot of reptiles nowadays is, because of global warming, it depends on which type of reptile you're talking about, but you might see a huge increase in males in the case of leopard geckos, or a huge increase in females in the case of many turtles, which in turtles it's kind of the opposite. If they have high temperatures, it's females that hatch, and low temperatures, males that hatch. What you needed for the albino trait to appear is that you needed to have a female who was heterozygous. So she had the big D for dots because she was able to make melanin, and she had a small d for albinism. You also needed a male with that same trait. Now, according to the law of independent assortment, the law of segregation, according to the law of segregation, her gametes, this is on one chromosome, this is on another chromosome. So this pair of chromosomes is what carries the big D and the small d. When meiosis happens and these alleles separate, some of her egg cells contained a big D, some of her egg cells contained a small d. The male, some of his sperm cells contained a big D, some of his sperm cells contained a small d. This was the process in order to make those sex cells, which would come together. So one chromosome carrying either one of these alleles would come together with a gamete that had one of these chromosomes, so the chromosomes could pair up, because leopard geckos are a diploid organism. We're going to do a Punnett Square here, which in this particular case we're looking at one allele. So mono, hybrid is when you're breeding 2 things together. So monohybrid cross is when we're looking at one allele. The Punnett Square, as I said, the male would be on the top. The male contributes either a big D or a little d, depending on which chromosome he ends up passing on to that gamete. The female also has a big D or a little d, according to whichever chromosome she passes along in her gametes, in her eggs. When they meet, you have a possibility of a big D and a big D and a little d, big D, little d, and then a little d. So the question would be, what percentage of the offspring would be albinos? You have to be homozygous recessive in order to see the albino trait. This would be a wild type normal colored. This would be a wild type normal colored. This would be a wild type normal colored. This wild type would look exactly like this wild type because it's a dominant trait. There's nothing you would be able to see in the phenotype different between these individuals. You would only see the difference when we have the albino. And that's the homozygous recessive. Heterozygous and homozygous dominant would look exactly the same. If we look at another allele, I just pulled out another one that I've actually bred over time, the blizzard allele. The geckos in a blizzard lizard or a blizzard leopard gecko don't have yellow coloration in their skin. They also don't have dots, but the more important part is that they don't have the yellow coloration, which you would see in a normal wild type. The dots are also important. But this is another recessive gene. And in this particular case, we're doing a dihybrid cross. We're looking at two separate alleles. I take my blizzard lizard, my lizard who is homozygous for the blizzard characteristic, which I'm going to call little b. He's a little b, the recessive form of the blizzard trait. And we'll just assume that since he does have some darker grayish color on him, he would be big D as far as albinism is concerned. Whereas an albino would have probably the dominant form of blizzard, because it's definitely not a blizzard lizard. So it does the dominant, not blizzard gene, while it also has the little d for being an albino. Now, when we cross these two, they're going to pass on the father's going to pass on a big D and a little b. The mother's going to pass on a little d and a big B. That's all she has to pass on because she's homozygous recessive for albino. She's homozygous dominant for not blizzard, whereas the blizzard lizard is homozygous dominant for not albino for a more of a dark coloration, and then little b for the blizzard. So all of the offspring are going to get this big D from the father. They're going to get this little d from the mother. They're going to get this big B from the mother, and they get this little b from the father. So they're heterozygous now for both albino and blizzard. That's not exactly a surprise, I don't think, but it might be interesting to see that you cross these two lizards, which both have recessive traits, but they also have dominant traits. And when you combine these dominant traits with the recessives, actually the dominants take over, and you get a very normal-looking leopard gecko. If you crossed an albino with a blizzard lizard, you wouldn't really be able to tell that it's not a normal leopard gecko. It's only when you cross the first generation. Now, again, this was done with leopard geckos. They bred brothers and sisters together. I know that's usually not a great idea. Lizards, reptiles, you can do it for a generation or two, and it doesn't cause major problems, which you would see in humans in many cases. And then we do the Punnett Square. All of the offspring are going to be, as we said earlier, big D, little d, big B, little b, meaning they're heterozygous for albino and they're heterozygous for blizzard. They would look perfectly normal as far as their coloration. But when you cross them, you have to look, what can dad pass on? Dad can pass on big D and little b. Big D and big B. Dad can pass on. So this is the law of independent assortment, where the D and the B both get passed along. They're not connected together. You wouldn't have a big D connected to a big B. The D can assort with the B in any way. So the big D can assort with the little b because these are on two separate chromosomes. The little d can associate with the big B because they're on two separate chromosomes. The big D can be with a little b, or the little d can be with a little b. That's what the father, these are all possibilities of what the father might pass on. And the mother, since she's also heterozygous for these characteristics, she would have the exact same combinations of chromosomes that she can pass on to her offspring. I'm sorry, I get my 9 wild type, I get my 3 blizzard, I get my 3 albino, and I get my 1 blizzard albino. There it is. Finally, it all's working together. There are a couple of other traits I just wanted you to get introduced to. Incomplete dominance is where you have maybe something like wild type and something like super snow in the homozygous state. You've seen what the wild type looks like already. This is what a super snow would look like. If I cross a super snow with a wild type, I get something kind of in between. The wild type has a lot of yellow coloration. The super snow doesn't have any. So I get kind of a yellowish white color. If you remember my leopard gecko Rocco, that's what he has. So wild doesn't win, super snow doesn't win. Rocco has a combination of the two features. That's incompletely dominant. One can't win out over the other. They end up kind of coming to an agreement where they are mixed in terms of their combinations. Codominance is where we see the effects of each allele, but it's kind of each one is expressed in its own way. Therefore, if I have, for example, these are a kind of flower. These are begonias, I believe. So I have a red one, I have a white one. If I cross the red with the white, if it's incompletely dominant, they're going to be pink. There's going to be a combination of those two features, just like I had the yellowish white here. But if it's codominant, I actually do see red and I see white both expressed. If you cross a white cow and a red cow, same thing. You get a roan cow. Some of it's red, some of it's white. It's codominant. Both dominant features are being expressed. When I take the wild type ball python and I combine it with the super fire ball python, I end up getting something that has some of the normal coloration of a ball python, but I do see a lot of the white here because of the super fire being expressed. So codominant, in different areas, you see different parts of this being expressed. It's not a blend of the two, which we would see in incomplete dominance. It's actually the red and the white, or the red and the white, or the white and the brown are actually being expressed in the organism. Sex-linked inheritance is only in species where you see sex chromosomes. When I talk about leopard geckos, as I said, their sex is not determined by chromosomes. In mammals, however, most mammals have an X chromosome, and then if they are a male, they have a Y chromosome. If they're female, they have 2 X's. The X chromosome codes for a lot of important genes. The Y chromosome, very few genes. Very few alleles are on the Y chromosome. So if you have some sort of a chromosome if you have some sort of an allele for a trait on the X chromosome and you're a male, that's the trait you're going to have. If you're a female, you might have a trait such as color blindness, but you also have the allele, maybe a traditional color vision on the other allele, which means that one would be dominant. A lot of these sex-linked traits are recessive. Color blindness would be recessive to a gene to be able to tell the difference between red and green. So if you're female, you might have the recessive color blindness gene, and then you have a red-green able to see gene, which means that you're able to see colors. However, if you're a male, if you have the color blindness gene, then you're going to be colorblind. This is the same thing true with hemophilia. It's a disease about clotting factors in the blood. At least 2 of those genes are on the X chromosome. If you're a boy and you have a hemophilia gene, you're going to have hemophilia. If you're a girl and you have the hemophilia gene, you also may have a normal blood protein gene on this X chromosome that can cover up it can be dominant to the hemophilia gene. We see this a lot in terms of inheritance. So if the father doesn't have this characteristic, the mother might have it hidden because hers would be recessive. In this particular case, you could be talking about color blindness or hemophilia, which would be masked, would be hidden because the woman has the dominant allele, which would code for blood clotting factor or would code for being able to see color. The mother would pass one X chromosome on in each of her gametes. So in this particular case, one got passed on to this girl, one got passed on to this girl. The father can only pass on either an X chromosome or a Y chromosome. If he passes on the Y chromosome, then chances are his child will be a male. If he passes on the X chromosome, chances are his child will be a female. It's really what the male contributes in his gametes that determines what the children would be. What you can see here is because mother can distribute one of two possible X chromosomes, one which has maybe color blindness or hemophilia on it. If she passes that X chromosome and dad passes the Y chromosome, that child would be affected by that gene. Whereas if she passes along the dominant version on her other allele and dad passes on the Y chromosome, this child's not going to be affected. Girls could be carriers, but they wouldn't necessarily show any effect from that gene, which might not be expressed in her case. And then we have some much more complex sorts of genes. So this is a particular one called carrot tail. We don't have one gene that would give leopard geckos a really nice carroty looking tail. It's actually a combination of genes, but you just kind of breed them and breed them and breed them, and if enough genes line up, then you end up getting more and more of this orange on their tail. A lot of diseases are very complex. We will talk about some simple inheritance genes that are related to certain diseases like sickle cell or like hemophilia is another example, but a lot and a few other diseases, but many diseases have much more complex inheritance patterns, which is not just through one allele or one gene. Many of our articles that we read were about genetic engineering, so where does that fit in? Genetic engineering is used to express a trait that does not naturally occur in the species. This is where we are putting alleles into a species that doesn't normally exist. They could be dominant, they could be recessive. If you remember the gene drive, that's a very dominant gene. So what that does, if it's inherited by the offspring, it will render all of the males sterile. The males will not be able to reproduce, and a species can die out very quickly for that. If you remember the article about wiping out invasive species on islands like the Galapagos where turtle eggs are being eaten by rats and mice, we want all of the males to go sterile. And if you have a dominant allele like that, it can happen very quickly. CRISPR-Cas9 has made genetic engineering much, much easier. This is a very new technology, but it is actually making genetic engineering much easier to swap in a gene that we might want. That's the actual system. So what we're doing, we haven't looked too much at the actual structure of DNA yet, but we are cutting DNA that we want to get rid of. We're putting in DNA that we want to keep. This is how the system works. We swap out a gene that we don't want. We swap in a gene that we do want. Now, what are some examples of those genes? In fish, we might be putting in a gene to make them glow. This is the glowfish. This has been available for maybe 10 years or so now. It's actually a jellyfish gene that causes these fish to glow. So we take a gene out of jellyfish, a gene that we want to be in fish. We swap out a little bit of DNA and we put in this fluorescent protein, which was originally a green fluorescent protein, but now we've been able to alter the gene sequence a little bit, which changes the protein sequence, and we have many different colors. Another case for this is making plants that are resistant to herbicides. Roundup is an herbicide used to kill plants. I'm sure you've been exposed to it at some point. Someone's been spraying it somewhere to kill off weeds, as we call them. What you do with Roundup Ready is you have a plant like soybeans. It's very resistant to Roundup. It's been genetically engineered to not get killed by Roundup. You spray Roundup on the field. The only stuff that survives is the Roundup Ready soybean. All of the other plants would die off from this. Now, we don't exactly understand very clearly what the health effects are of this on humans, although we do know that Roundup is very bad for amphibians. It's been documented that it's a terrible killer of amphibians, frogs, salamanders, things like that. We've also been genetically engineering pigs that might be resistant to certain kinds of diseases. We've been genetically engineering pigs that the males will not mature because that changes the flavor of their meat. A lot of genetic engineering is happening in terms of our food, and a lot of the foods that we eat have been genetically engineered. And that was one of the articles we discussed, golden rice, which this was very relevant, and so was the discussion about it. I always love to bring people back to what Dr. Malcolm says in Jurassic Park. People are so obsessed whether they can do something, they don't think that they should. But we're moving into an era where a lot of these decisions and a lot of these techniques are being used, and we haven't really decided whether we should be doing them or not. Now, some things are useful. Another example would be we're actually putting human genes into pigs. Pigs have organs that are about the same size as a human. If we could put enough human genes into a pig to allow a pig's organs to be transplanted into a person, now you have a very easily accessible source of organs for people who might need new hearts and new lungs and new kidneys. It's less of an ethical issue to kill a pig and harvest its organs than it would be to do that to a person. The question is, though, if we're putting human DNA into pigs, at what point do we stop considering them pigs, and what point do we start considering them humans? These are all questions that don't have answers yet, and I just wanted to kind of get you thinking about this, because this is the world that you are going to be entering. One other way that we've been talking about CRISPR and genetic engineering is to bring back endangered species. We talked about the woolly mammoth. The woolly mammoth, we would put in woolly mammoth genes into Asian elephant cells. We would get those Asian elephants to grow up, and from there we would we would put those Asian elephant cells with the woolly mammoth genes into a mother. It would develop, a woolly mammoth would be born. This is another example of this. This is the passenger pigeon, which is a bird that went extinct about 100 years ago now. And what they have talked about is taking passenger pigeon genes, putting them into a closely related species, get those genes to the sex cells of these birds so that when they mated, they would actually have passenger pigeons as their offspring, or something that looks very much like a passenger pigeon and acts very much like a passenger pigeon. It's a possibility to bring back species that humans have made extinct. Another thing that might be happening in your lifetime, and I just wanted to make sure that you were aware of it. That concludes this slide presentation. It's a little bit long. I'll edit it down, and I hope you get out everything that you need for the upcoming graded discussion.